for those of you I haven't met before, which is most of you, my name is Dionne. I'm a faculty member in material science and engineering at Stanford. I also have a courtesy appointment in the School of Medicine in the Department of Radiology. And I think we have about 2 hours together. I actually just flew in this morning. My flight was supposed to land at 10, but it got a little bit delayed. So don't be shy to ask any questions because, yeah, I'm a bit jet lagged, and it's very possible that you know, I'm not clear because of that. So don't be shy to jump in and ask questions. I'm also not used to giving classes sitting down, but I guess since they're recording the Masterclass, they need to have this particular view. So yeah, bear with me. So I wanted to share with you some of the work that my students have done at Stanford in developing basically optically coupled transmission electron microscopy to try to understand how catalysts work at the atomic scale, and then linking those insights to kind of reactor scale performance of these catalytic materials in a variety of different catalytic reactions, but most of which at least involve some sort of hydrogen and hydrogen splitting to be added to another molecule, whether in acetylene hydrogenation to create ethylene, or in ammonia synthesis, so taking N2 and H2, or in kind of more green like steel manufacturing. So those are kind of the 3 example reactions that we'll focus on. I also, throughout the kind of masterclass, will have pictures of students who contributed to the work. You'll see many of the folks listed here are lab alum, including Andrea Baldi, who is actually here now in the Netherlands as a group leader at VU Amsterdam, Vrije University. So first, just a quick introduction to my lab at Stanford. What we're really passionate about is being able to sculpt light at the nanoscale, both to detect molecules and then also to direct their transformations. And my understanding is that many of you are interested in photonics and nanophotonics. Later on in the week, I'll be able to give a presentation at the NWO conference, and I'll share some of our work on detecting molecules. But to start, I wanted to just share a bit about me besides my academic upbringing. So I was born on the East Coast of the United States in Rhode Island. And for those of you who haven't been there before, it's typically pretty cold and snowy in the winters. So I grew up figure skating and thought that that would be my professional career. I don't get a chance to do it too often now, but my students still entertain me by going either disco skating or roller skating in this particular case. My interest in science was inspired by Special Agents Fox Mulder and Dana Scully. I don't know if anyone here ever watched the X-Files or has heard of the X-Files, but I thought it was pretty cool that basically a scientist and a physician could work together as a team to solve mysteries on a daily basis. So after I wanted to become a figure skater, I thought it'd be really cool to be a paranormal researcher. And this was back in like the 80s when brick and mortar bookstores used to be pretty common. So I started going to the bookstore and reading a lot about the paranormal. And thankfully the PA section was right next to the PH section. So I started reading about physics and photonics. And I think from an early age that got me kind of fascinated in like the you know, mysteries, especially of the quantum world. And then for my PhD, I got to move to California, where I did my PhD in applied physics at Caltech and got introduced to nanoscience and nanophotonics. And I got to meet some pretty cool science stars along the way, including Stephen Hawking, and then also the Mythbusters guys, Jamie and Adam. So it's been a ton of fun being a scientist, and since many of you are PhD students, I'll say the best is yet to come. Like, hopefully you've really enjoyed being a PhD student. I know sometimes the days can be long, and it's tough if an experiment isn't working out, but hopefully you'll find that the integral of your experience is positive, and I think you'll find that life just keeps getting more and more interesting, especially like the more you learn. So I just wanted to put up one image that's a bit of a preview for the presentation I'll give on Wednesday. What my lab has done is try to develop nanoscale and nanophotonic materials that can control light on the cellular to molecular level to be able to detect molecules, including nucleic acids, metabolites, and also to detect cells. And in particular, we've been especially excited to use vibrational scattering and Raman scattering to be able to identify cells' antibiotic susceptibility. Basically, we've developed not only the nanoscale materials, but also some of the ML models to be able to provide interpretability to the Raman scattering spectrum. Okay, so for today's masterclass, hopefully many of you are passionate, not only about physics, but also about sustainability. And if we think about the challenges that the world is facing in sustainability, there are many, but the ones that our lab is really focused on is trying to look at either production of chemicals and how we can do that in a more sustainable way, or production of plastics and like consumer materials, or production of like building and construction materials. And collectively, those three, so chemical production, plastics, and steel, account for about 15% of all global greenhouse gas emissions. And that's because for all of those materials, whether you're creating, say, fertilizers, which you have to do to be able to feed the world, or if you're trying to create, say, pharmaceuticals, which require a catalyst, or plastics, usually you're going to use a catalytic material. And catalysts generally have to operate at really high temperatures to be able to enable the chemical conversion from whatever your reactants are to whatever the products are. So generally, catalysts are operating at high temperatures, so that's one pretty big problem. Another challenge that I'll share with you in a few slides is that most catalysts are not selective. So you can almost think about light as kind of a chemical sledgehammer. You're kind of taking your reactants and trying to get them to get to the product you want, but simply by heating up your reaction vessel, you're actually getting a lot of byproducts as well that may not be as advantageous. So you generally don't get product selectivity when you're operating with traditional thermal catalysts. So we've been exploring how light as a more sustainable energy resource can enable basically product selective and high yield catalysis. And the cool thing about light is that you can drive systems out of equilibrium. So you might remember from some of your undergraduate classes that basically thermal equilibrium means when you're in the dark, right? You don't have any excited states. And light is basically generating electron-hole pairs and hot electrons and hot holes that can open up entirely new reaction pathways, like excited state reaction pathways, that are not accessible with traditional thermal catalysis. Okay, so we'll start first of all with what a catalyst looks like. And there are two main classes of catalysts. The first is kind of an enzymatic catalyst, or like a protein. So that's more of a biological catalyst. And our body basically is, synthesizing, essentially new materials like every second. Like we're able to, go from DNA, our genetic code, to RNA, which then gets basically transcribed into proteins. And a cell has roughly over 1 million proteins that are all assembled, and in many times by like these enzymatic catalysts. We won't focus in this presentation too much on these biological catalysts, but a lot of the techniques that we'll discuss have some relevance and some parallels to the enzymatic catalysts. There's also metal nanoparticles, which are more inorganic nanoparticles. Kind of the advantage of these metallic nanoparticles is that we can synthesize kind of a broader range of either monometallic or multimetallic nanoparticles, essentially using the entire periodic table as our palette. And we can tune the size, the shape, the composition, the phase, the surface faceting, basically to be able to get more tunable catalysis. And this is unlike the protein case where basically there's just a few elements. Oh, yes. Oh, is that what's happening? Oh, yeah, perfect. No worries. Thanks. I was wondering what was hitting it. So enzymatic catalysts basically have basically well-defined like structures and very localized structures where the molecules can interact. And that is kind of convenient if you want very like predictive chemical transformations to occur. And if you compare that with metallic nanoparticles, electrons and photons are essentially probabilistic in nature. And there's currently not enough understanding of how charge carriers move through the diverse inorganic metallic nanoparticles to provide a complete picture of rational design principles. And this is a quote that I had found from Nishori in Chemical Catalysis, published just a couple of years ago. So I think it's neat to think about using the tools both from inorganic chemistry to be able to create new metallic catalytic nanoparticles, and also from synthetic biology to be able to create new enzymatic catalysts. But I think there's kind of a much richer, albeit less understood, kind of toolbox to play with when we're dealing with these metallic nanoparticles. And in this presentation, we'll focus mostly on these metallic nanoparticle catalysts. And essentially, as you shrink down the size of a metal nanoparticle, that's when the catalytic activity gets much stronger. So this is a plot from Jens Norskov, where he was determining the catalytic activity for CO2 reduction as a function of the diameter of the gold nanoparticle. And many of you know gold is a noble metal, which means it's normally not reactive, right? So in bulk, you're not going to get many reactions that occur on the surface of the gold. But as you shrink down the size of the metal nanoparticle to below just a few nanometers, you can start getting this almost exponential increase in catalytic activity, regardless of which substrate you're on. So regardless of which substrate you've put the gold nanoparticles on. So why don't we just shrink down the size of the metal nanoparticle to improve catalytic activity? Well, it's because as you start reducing the size, even though your catalytic activity goes up, you have to think about the structure and facet dependence of the nanoparticles as well. So here's an example that shows basically what the catalytic activity is for formic acid oxidation on nanoparticles of different crystallinities. So if you have a cubic nanoparticle, in this particular case, you wind up getting the highest catalytic activity for this particular reaction. If you have an octahedral nanoparticle here, even though it has the same number of faces, because you have that 111 termination instead of the 100 termination, you get much lower catalytic activity. And then there's also composition dependent. So as you start thinking not just about monometallic catalysts, but bimetallic structures, you can see that as you change, for example, the concentration in this case of silver on palladium, you can start to boost the selectivity for the particular reaction that you're looking at, but the conversion or the yield of the product that you want to get actually goes down. And this reaction is actually ethylene formation, which is basically a precursor for polyethylene, one of the most commonly used like consumer plastics. So basically, if you reform naphtha, so basically if you take these oil reserves, you'll have acetylene, and you want to hydrogenate acetylene to get ethylene, right? So basically you're breaking this C triple bond C. You're adding in hydrogen gas, and some of that hydrogen can get added onto the acetylene, basically to form ethylene, which allows you to create polyethylene. But unfortunately, usually not all of your acetylene gets converted, and you can overhydrogenate it to form ethane. And overhydrogenation is not the worst thing, but basically, like if you have trace acetylene that doesn't get properly hydrogenated, you wind up not forming the plastic. So basically, the trace acetylene winds up being a poison to the downstream polyethylene formation. And you wind up not basically getting the product you want. So you want to avoid overhydrogenation because basically ethane, just a gas, that's going to go away, so it's not the product you want. You want to get ethylene, but you also want to make sure that it's almost 100% conversion into the ethylene. And then the same goes for plastics upcycling. Just an interesting fact is that I think roughly about a dump truck worth of plastic is put into the ocean roughly every minute, and that's led to this like great Pacific like garbage patch where you can see just plastic kind of floating in the ocean. And then of course like that plastic gets broken down into microplastics, which are having pretty severe consequences on our ecosystem. And it's actually pretty challenging to recycle plastics. You can either try to mechanically grind the plastic or you can try thermal processes, but usually in any of those recycling processes, you wind up getting kind of a large distribution of these N-alkane chains, so you don't get a controlled distribution in your plastics upcycling. So ideally what we'd like to do is use light to be able to create more selectivity in reactions like this. So that way, for example, you could get a very sharp distribution of N-alkanes in your plastics upcycling and also get, pure ethylene. So this would allow for a more circular economy. So here's where photonics comes in, or I guess before I get into light, any questions on catalysts? Go for it.
I want to know how does Facet decide the kind of product which will be formed? How does basically how does Facet give you a particular product?
Oh yeah, amazing question, and I'll show you some of our work using transmission electron microscopy that basically kind of shows that facet dependence for hydrogen splitting. But basically, like what you'll see in some of the videos is that the low coordination number sites matter a lot for catalysis. So basically, whenever you have kind of sharp tips, that's where the molecules tend to be splitting kind of with the highest rate. So you get a lot of kind of kinetically preferred activity at those low coordination numbers or low coordination number sites. And basically the faceting determines like what precisely that low coordination number site looks like. And then it also determines, as you'll see in some of the hydrogen splitting, how the hydrogen intercalates into the surface and the subsurface, basically, of some of these catalytic nanoparticles. And what we'll see is that, like, especially in these hydrogenation-based reactions, you want to have splitting of hydrogen, but you also want to have a little bit of a reservoir of hydrogen that's kind of in your nanoparticle. And that surface faceting will determine basically how deep and how far the hydrogen goes into the nanoparticle. Yeah, it's a really great question. Any other questions? Okay, so we'll dive into using light to control catalysis. So generally the knobs you can use to tune your kind of catalytic transformations include pressure, right? You can change the pressure of the gas or the chemical potential of the gas. You can also turn up the temperature, which is what we use generally in thermal catalysis. You can also do electrocatalysis, where you apply an electrical bias, or you can also do photocatalysis, where generally you're applying light, but you're not necessarily controlling it at the nanoscale. And all of these properties are basically macro scale, so much, much larger than the size scale of your catalytic nanoparticle, which is typically just a few nanometers or tens of nanometers across. So I think the exciting thing about plasmons is that they give us this ability to control light on the nanoscale. And I'm just curious, who here does research in plasmonics? Okay, cool. So not everyone. So I'll just spend a minute on this slide. So you know that light is an oscillating electric and magnetic field. And if for the time being we just think about the electric field of light, when that interacts with a metallic nanoparticle, it basically causes the free conduction electrons to oscillate back and forth in response to that driving electric field. So you wind up getting this cloud of electrons that gets basically displaced, in this case up and down, to match with the polarization of the electric field of light. So for the small nanoparticles that we'll be dealing with in this particular presentation, we basically just have a dipolar-like resonance in our catalytic nanoparticles. And here, if we just focus on that lowest order of resonance, so the lowest energy resonance, like that dipole mode, these plasmon resonances can be like readily spectrally tuned depending upon the size and the shape of the nanoparticle. So as you increase the size of the nanoparticle, generally your dipolar resonance shifts to longer wavelengths, and that kind of makes sense, right? Because it takes longer for your electron cloud to slosh back and forth, or if you think about your dipole moment as q dot d, right? Basically your distance is larger. And then here's an image actually taken from some of my students using a transmission electron microscope, where they mapped out different plasmonic modes of a flat kind of prismatic nanoparticle. And you can see that basically the nanoparticle can support different types of plasmon modes. So here at about 3 electron volts, there's kind of this tip mode where the electrons all want to concentrate around the tip of the nanoparticle. There's also at slightly higher energies this edge mode where the electrons kind of oscillate around the edge. And then there's also kind of a bulk plasmon mode, which is essentially the metal to dielectric transition. It's almost like this kind of symmetric breathing mode of the electrons in the nanoparticles. And I have a bunch of references here. The talk is recorded. I'm also happy to share some of the references or slides if you'd like them afterwards. So throughout the past handful of years, many people have shown that these plasmonic catalytic nanoparticles can allow for significantly increased catalytic activity. And here's just one example from Sujo Linich's lab at Michigan, where he also was looking at CO2 reduction, and basically like at the same temperature as thermal catalysis, when he turned the light on, he could see increased catalytic activity. And you can imagine the question, you know, when papers like this first came out, is like, to what extent is heating playing a role versus to what extent might hot carriers play a role, and basically just photoexcitation of electrons kind of being injected into particular HOMO-LUMO orbitals to drive chemical catalysis. So the paper that I think kind of opened up the field to the idea that it wasn't just light causing more heating was some work by Dane Swear and Naomi Hollis and Peter Norlander, where they compared the selectivity and the conversion or the yield with thermal catalysis and with photocatalysis for that acetylene hydrogenation reaction that I shared with you earlier, basically the one that creates ethylene for polyethylene. So you want to have ethylene, you don't want to have ethane, the fully hydrogenated version. And what they found is that when they were looking at thermal catalysis, they could see basically as they increased the temperature, the yield for ethylene went up, but the selectivity started going down. So as they went to higher temperatures, they basically wound up fully breaking that C triple bond C and basically wound up with the fully hydrogenated ethane. Whereas with photocatalysis, as they turned up the laser power density, the selectivity went up, and the conversion basically kind of flattened off. So if nothing else, you can see there are different trends between thermal catalysis and photocatalysis, not the same trend, which indicated that there was basically some other mechanism going on. And to better understand what those mechanisms are, you really need to think about a variety of length and time scales. So at the smallest of length scales and kind of the fastest of temporal scales, you can think about illuminating a metal nanoparticle, here shown basically just as this gray quadrant of a sphere. And then also kind of superimposed on it is the Fermi-Dirac distribution of electrons. So as you are photo-exciting your carriers in the metal, you basically wind up with a thermally excited, or like a optically excited distribution of carriers that basically is hotter than the thermal distribution than you would expect, right? So you have your normal Fermi Dirac distribution, but photons are now exciting more carriers above their current, occupancy or current state, and those are hot carriers that basically can be rapidly injected into various either homolumal levels or vibrational modes of the adsorbates and molecules that are in the vicinity. So there's the possibility to use these hot carriers, and in some cases hot holes also play a role. Then there's also that near field enhancement. So just by virtue of having extra conduction electrons, you not only have more hot carriers where you have these electromagnetic hotspots, but you also have potentially local heating effects, and you also have increased local densities of optical states that can basically change your transition rates between your HOMO and LUMO levels. And then to the question that got asked earlier, like how does the faceting play a role? Depending upon the molecules that are reacting and the catalyst you have, there's also site selectivity. So you have to think about where these electromagnetic hotspots are with respect to where the catalytically active sites are. And then as you zoom out to larger length scales and kind of longer time scales, there's collective heating effects and electromagnetic hotspots that can be distributed between the nanoparticles that are in your catalytic reactor. And those localized kind of heating effects can give rise to macroscopic thermal gradients. So you can think about this as being a cross-section through your reactor. And if you're sending a light in from the top, you can have heating that basically gives rise to kind of a non-uniform distribution of temperature within your reactor. So we kind of need to think about all of these processes when we're designing reactors. And what I'll do in this presentation is basically start with some of the basically faster and smaller length scale processes, and then we'll work our way up to more of these like bench scale reactor processes. So for the smaller length scales, we use transmission electron microscopy. And when I first joined Stanford, I was lucky enough to join right when the Titan transmission electron microscope was being installed. You can see an image here. Basically, you have your electron gun, and then the system is like differentially pumped, which means that the gun is basically under vacuum, and as you get closer and closer to your sample holder, basically you start approaching atmospheric conditions. So we have all these reactive gases that can be introduced basically to the sample, so that way we can do environmental transmission electron microscopy. And it's a pretty cool setup, like basically going from high vacuum to kind of atmospheric pressures of gases is quite an engineering feat, and I definitely give the engineers credit for being able to enable that. And then not only do we have this reactive gas environment, but we also have the ability to heat the sample, cool the sample, bias the sample electrically, or we can also send light into and out of the transmission electron microscope. So we worked closely with Gatan to be able to create basically an optical holder where the sample in your transmission electron microscope is surrounded by two parabolic mirrors. And then we have fibers that allow light to be coupled into and out of the TEM. Has anyone here done like photoluminescence? Just out of curiosity. Okay, only a couple people. And that was in an SEM or a TEM? SEM. Okay, neat. So it's pretty similar, except in a TEM you only have like a 5 millimeter pole piece gap. So the tools basically are quite a bit smaller, and the space with which to get optics into and out of the TEM is quite a bit more confined. So I think when you look at the landscape of plasmon photocatalysis, there are kind of two key classes, one of which is antenna reactor systems. So this is a little more similar to what you might find in certain biological systems like photosystem 1 and 2, where most catalytic nanoparticles are intrinsically not the best light absorbers. Like they're very chemically reactive, but they maybe are not very good light absorbers at visible wavelengths. especially if you want to use sunlight. So what you can do is basically put your catalytic nanoparticle, in this case, for example, palladium, near another metal that is a good light absorber. So gold, for example, is a very good visible light absorber. Silver is another good visible light absorber. There's a second class where you basically take both of those and alloy them together. And these are essentially bimetallic alloyed nanoparticles. And here you can imagine there's a whole suite of properties that can be tuned, like the composition. And I think as you look through the literature, you'll find that these like single atom alloys are gaining like a lot of popularity, because there's a lot you can do by like tuning the position of a single atom or just a few atoms on these nanoparticles to be able to tune the catalytic activity. So in the presentation today, we'll focus on basically silver and gold as the light absorber, and then palladium as the catalyst, and we'll look at kind of both of these sorts of nanoparticles. Okay, so a lot of catalytic reactions have hydrogen involved, basically like that hydrogen molecule will get split into two hydrogen atoms or ions that then can be added onto another reactant. So these are generally kind of reduction reactions. And what we were interested in a few years ago is studying basically how hydrogen was interacting with some of these plasmonic catalysts, basically to use that then to be able to control like some of those more complex chemistries that I mentioned at the beginning, like acetylene hydrogenation or steel manufacturing or ammonia synthesis. So basically when hydrogen interacts with a palladium nanoparticle, it splits into two hydrogen atoms and then can start intercalating into the palladium nanoparticle. The palladium is kind of a sponge for the hydrogen. And that can be both good and bad. In certain cases, it can be bad because if the palladium absorbs too much hydrogen, basically you wind up with lower activity, so basically lower catalytic activity, and here it's shown for CO2 reduction. So basically as you get to higher and higher pressures of your hydrogen, you can see that the catalytic activity, the slope here in these dashed lines, basically starts to get reduced. And that's because as you get to higher pressures, more hydrogen is intercalating into your nanoparticle, and you're able to use less for that CO2 reduction. Here's another example, again going back to the ethylene formation, where as you get to higher pressures, basically you wind up forming more of that undesirable fully hydrogenated product, that ethane, and less of the ethylene, that not fully hydrogenated product. Okay, so what we did is use optically coupled transmission electron microscopy basically to probe how the hydrogen was intercalating into these palladium nanoparticles. And basically when hydrogen splits on the surface of palladium, it starts intercalating into the interstitial sites. So here each dot is basically a palladium atom. Hydrogen is in the surrounding. It starts to intercalate into these interstitial FCC sites. And then when you have a high enough hydrogen pressure, high enough chemical potential, basically the nanoparticle or the lattice expands, and the lattice expansion is about 10% in volume. So it's a pretty enormous lattice expansion. So we call these two phases the alpha phase, which is basically the hydrogen poor phase, and the beta phase, the hydrogen rich phase. So we can capture when hydrogen is going into the nanoparticle using electron diffraction. And I think many of you are physicists in the audience, so you're probably pretty familiar with diffraction. But basically here we're looking in K space, or like momentum space, how the electrons are diffracting through the nanoparticle. So when we go from the hydrogen poor phase to the lattice expanded hydrogen rich phase, we get a contraction basically of that diffraction image.
May I ask?
Oh yeah,
So
please
the
do.
difference between these two phases are only the concentration of hydrogen.
That's right.
And the existence of hydrogen doesn't influence on the structure of the original PET structure?
Yeah, great question. So the main structural change, and you'll get to see some videos of this too, is just that lattice expansion. Yeah, so there's not a structural phase change apart from the lattice expansion. Yeah, it's not like you're going from FCC to BCC or some other like weird phase.
Yeah.
Yeah, great question. And then in addition to looking at diffraction, we can also look at spectroscopy. So in our TEM, we have 300 kiloelectron volt electrons. They basically are kind of, you can almost think about them as like rain droplets coming down onto the nanoparticle. And then many of them kind of pass through without interacting with the nanoparticle at all. But sometimes the electrons lose energy basically to the nanoparticle itself, or gases in the environment, or the various electronic modes of the nanoparticle. So here's an example where we have hydrogen in the environment, and you can see some of those 300 kiloelectron volt electrons have lost, and this is the electron energy loss spectra, 12 electron volts simply by passing through the hydrogen gas. And that's what you would expect basically when you think about you know, what is the ground state of like the H2 molecule. So it's pretty close to what you'd expect for electrons basically exciting homolumo levels in H2. And then you have some electrons that are losing energy basically to the various plasmonic modes in the nanoparticle. And in this case, we're looking at somewhat higher energy, so this is the bulk plasmon mode. So as we go from no hydrogen, kind of shown here in purple and blue, where we get that plasmon mode or bulk plasmon mode at just under 8 electron volts, to hydrogen in the environment, there's about a 2 electron volt shift basically in that bulk plasmon resonance. So we use a combination of both EELs, electron energy loss spectra, and also diffraction to kind of probe how the hydrogen is intercalating into the nanoparticle. And we started off looking at this phase transformation because, as you can imagine, it's kind of difficult to follow chemical catalysis with molecular resolution in the TEM, and we have some techniques that we're working on now for that. But this was a cool way to figure out at least what hydrogen was doing in the palladium nanoparticle and at the surface, and then we could use that basically to infer how the hydrogen would interact with other molecules in the environment. So the first project that we did was now almost 10 years ago, where we looked at single crystalline palladium nanoparticles, and here are palladium nanocubes of different sizes. This was work done by Andrea Baldi, who now is a professor at VU Amsterdam. And what we did first is just vary the hydrogen pressure and then look at whether the nanoparticle was hydrogenated, so 0% beta, or rather not hydrogenated, 0% beta, or hydrogenated, which would be basically all the way on the right-hand side here. So this is in the dark. And what we did is make sure that the system could reach thermal equilibrium, basically. With each pressure step, we basically waited about 10 minutes or so to make sure that like nothing was changing in the system when we collected basically the diffraction pattern to know which state the nanoparticle was in. So here you can see there's a very abrupt change from that alpha to beta phase. Also with the single crystalline prisms that are shown on the next row, you can see there's also a very abrupt change. So the system is either in the alpha phase or in the beta phase, but there's no coexistence between those phases when you're in thermal equilibrium. And then we also looked at these icosahedral nanoparticles. So here are 5 icosahedra, and you can see as we increase the hydrogen pressure, basically there's like more of a gradual transition. So when we looked at the diffraction pattern, it appeared as if it was like a more continuous change between the alpha and the beta phase. So to understand what was going on here, what we did is basically focus our electron beam down to a smaller probe size, so just about a nanometer, and then scan it over the nanoparticle. So when we're at low hydrogen pressures, when we had, say, like the electron beam in this pink position or the electron beam in this green position, we basically saw a peak at 8 electron volts, and remember that corresponds to the alpha phase, so hydrogen wasn't in the system. When we went up to high pressures, you can see that peak now has shifted over to just under 6 electron volts, so it's fully hydrogenated. And kind of regardless of what pressure we were at, we either found that the system, when we scanned the electron beam over the entire nanoparticle, was either in the alpha phase or in the beta phase. So it was either fully hydrogenated or fully dehydrogenated in thermal equilibrium. And this is what you would expect for like a conventional first order phase transformation. Kind of same with the prism nanoparticles that are also single crystalline, either fully dehydrogenated or fully hydrogenated when you're in thermal equilibrium. But with these icosahedral nanoparticles, we found something a little different. You can see, for example, here this pink dot and the green dot have kind of 2 peaks when you're at higher hydrogen pressures. So that means there are portions of the nanoparticle that are remaining dehydrogenated. And one of those portions is the core of the nanoparticle. And that's because these icosahedral nanoparticles are essentially not quite Platonic solids. You can think about the icosahedra as being composed of 20 tetrahedra that don't all fit together perfectly, so they need to expand a little bit on the surface and compress a little bit at the core to actually form that nanoparticle. So there's some compressive strain in the core of the nanoparticle that actually makes it much harder for hydrogen to get there. And remember, hydrogen sits at those interstitial sites. And then what we also found is that by doing tomography and dark field imaging and diffraction, we could basically map out those 20 tetrahedra within each individual icosahedra. And we wound up seeing kind of mosaic loading. So if one tetrahedra absorbs hydrogen, it expands, and that makes it harder for the neighboring tetrahedra to also absorb hydrogen. Okay, so that's kind of thermal equilibrium. The next thing was what's happening kind of in between, right? Or like when we're not in thermal equilibrium. And what we were able to do is start imaging how hydrogen intercalates into these systems dynamically. So you could see kind of from the contrast here that when hydrogen first came into the system, and I'll play it again. There. Whoops. I wanted to pause it for you, but now it's not letting me play. Let's try it again. There. So you can see hydrogen starts coming in through this corner. It looks like it's kind of diffusing along the edges, and then it starts coming in, eventually forming this flat interface, this 100 type interface, until it eventually kind of moves out of the system. So if we look at snapshots, what we were able to do, and you saw kind of the mouse moving throughout that video, is collect electron spectra during the video to be able to figure out whether the nanoparticle was in the alpha phase or in the beta phase. So here you see the hydrogen rich area. So hydrogen basically is coming in from those low coordination number sites, basically the tips. It forms kind of this diagonal interface, and then it looks like the hydrogen's kind of diffusing along the edges until eventually it kind of stabilizes along this 100 type interface, and then starts moving across the nanoparticle until it's fully hydrogenated. And if you're looking at this image, you'll notice that there's a little bit of a contrast change. Basically, this sort of image is taken by doing what's called dark field imaging. So we're looking at a peak in the diffraction plane, and that peak basically was moving in and out of like the aperture, basically as the system was being hydrogenated. So depending upon where that peak was with respect to the aperture, we either got the beta phase being bright or the beta phase being dark. And this was an experiment that we did, I think, back in 2017 at this point. So we've since worked out now how to kind of make sure the contrast remains consistent throughout the entire video. Yeah?
Sorry for interrupting. Sorry, it might be a very stupid question, as you mentioned.
Oh, no such thing.
Sorry. The beta phase was supposed to be physically large in scale, but it doesn't seem like it's present over here, or...
Oh, yeah. So here, the size of the nanoparticle is about 40 nanometers on an edge, so it's pretty big, so you can't really see that expansion here, but you can see it basically in the diffraction patterns. So here... yeah, it's about a 10% expansion in lattice constant, or lattice constant volume. So here, if you trace out where you expect the diffraction pattern kind of peaks to be, what we've done is zoom in to one peak, and you can basically see like right at the start of the transformation, the system is mostly in the alpha phase, but it does have some of that lattice expanded beta phase. Like you can see a little bit of the diffraction peak there. And then as the transformation has progressed, you can see more of the peak has moved to that beta phase. And then as it's progressed even further, you can see now there's more intensity centered on that beta phase, that lattice expanded phase, but it's also kind of spread out in like that radial direction. And then basically at the end of the transformation, the diffraction peak is starting to tighten up. So great question that led perfectly kind of into this slide. So you definitely are getting the lattice expansion. The interesting thing is you probably know that diffraction peaks, like the full width half max, basically corresponds to, in a way, the purity of the crystal. So you can see this kind of broad full width at half max here. So basically there are lattice imperfections that are forming when you have coexistence of alpha and beta, right? So basically your lattice is strained where you have the hydrogen going in, and that's giving rise to various imperfections that are kind of giving rise to this more spread out diffraction pattern. And here, just to be clear, we're zooming in just to that one diffraction peak, but you could zoom into any diffraction peak and see the same sort of behavior. But then the cool thing, I'll get to your question in just 1 second, is you can see here kind of at the end of the transformation, the full width half max is getting sharper again. And basically, like at the end, the nanoparticle, and you can't really see it so well on the screen, but it again is a perfect crystal. So I think it's kind of neat how when you're working with nanoparticles that are below a certain size regime, basically the defects can be pushed out. So the system can almost like self-anneal. And this is one reason why, like, when you think about lithium intercalation into battery electrodes, there's basically an isomorphic similarity between hydrogen intercalation in palladium and lithium intercalation into, like, say, nickel metal hydride electrodes in batteries. So generally, you want to work with nanoparticles below a certain size to make sure you can cycle them a lot, because then basically the defects get pushed out and you don't wind up having defects that make it harder basically to store the lithium or the hydrogen in this case. So everything we've done so far has been in the dark, but I think you'll appreciate in a couple of slides why it's nice kind of to have this foundation once we turn the light on. Yeah.
Yeah, it's very interesting to see how this phase moving and slowly transforms to beta phase from alpha. And I noticed that those two sides of images that you show, the transition started at different size of the crystal. One is from like the longer side, and this is from another side. So how did you actually inject the hydrogen into the system, and can you control that? So
oh yeah,
it starts?
Perfect question, and that's kind of precisely what you'll see. We can use light to control which tip the hydrogen goes in to. But otherwise, it's just like a probabilistic, right? So you can basically do this experiment many times, and there are 8 cube corners, and hydrogen is basically equally likely to load from any of them. So basically, as you cycle it, like you'll find that it basically is determined by kinetics and, like, you know, which site basically is the fastest to get the hydrogen to split there.
And if you use light, normally we can also focus the light into very small spot, maybe not as 40 nanometer, but I'm just thinking, so you start with one specifically on the corner of this, or actually it's large enough to cover half of it?
Oh yeah, great question. So the cool thing, and maybe I'll just go back to this slide that I showed at the beginning, you don't need to focus the light to a diffraction limited point. You can just rely on using the right wavelength of light to excite like different corner modes. Or if you have like an antenna reactor system or a bimetallic system, you can basically have just one of these, say, tips light up, and that becomes the site that's preferential. Yeah, great question. Okay, so you kind of saw also in those cube videos that hydrogen was starting to kind of diffuse along the surface in addition to entering at the tips. And here's some higher resolution images. Again, the projector doesn't really do it justice, but this was using a direct electron detector where we could get atomic scale images of the entire hydrogen intercalation. And what we found is that basically what happens is hydrogen kind of expands basically the entire subsurface of the nanoparticle. So it's as if once there's a tip that the hydrogen enters into, it diffuses along the first few monolayers, usually just the first like 3 to 4 monolayers of that palladium nanoparticle. And then basically there winds up being kind of a critical mass of hydrogen at one of the tips where then it starts kind of entering into the nanoparticle and forming, you know, this kind of flat phase interface like this, say 100 interface. So this will be important later on because what we'll show with light is that we're able to use this subsurface of hydrogen to be able to trigger its release with light at the right wavelength to be able to get more hydrogen when you need it, but basically turn it off when you don't need more hydrogen or when you don't want to overhydrogenate. And then since you were asking about the tips, basically we found regardless of what nanoparticle you look at, so whether it's the cubes or the prisms, or in this case it's an octahedral nanoparticle, which has those 111 terminations, we always get that new phase basically nucleating and growing at the tip. And then same here, like even with nanorods, you always get like the beta phase or the hydrogen going in through the tips. Okay, so now let's turn the light on. And we'll start off with the antenna reactor system. And we know that light is going to basically move the system out of equilibrium. So in this case, what we did is focus on palladium nanoparticles that are basically the catalyst. And then we have these gold disks that serve as the antenna to absorb light. And we lithographically patterned the gold disks. And then we had colloidally synthesized palladium nanocubes that we just kind of sprinkled on top. So we looked at a bunch of these antenna reactor systems, and here's one simulation of basically the gold disk next to the palladium nanocube with just about like a 3 or 4 nanometer separation between the gold that we were able to see. And here you can see there's like a strong electromagnetic hotspot basically in the gap between the palladium and the gold. So here's an example where you don't need to focus light to a subdiffraction limited scale. You can just basically get light concentrated to this really small volume simply by virtue of kind of these 2 coupled dipoles at the wavelength that you're exciting with. So here you can see the electric field is enhanced by about 40x in the gap between the antenna and or the light absorber and the catalytic nanoparticle. So what we did in these sets of experiments is basically, first of all, collect these isotherms. So basically we were in thermal equilibrium with the light off, and we wanted to know where does the particle fully load hydrogen and where does it unload hydrogen. And then we set the pressure just above the unloading pressure. So basically in the experiments you'll see next, the particle is fully hydrogenated, and then we want to see how light triggers the release of hydrogen from the nanoparticle. So here's a movie, the light goes on, nothing's happening to the diffraction pattern, but now you can see the lattice basically contraction, and basically that gave rise to a expansion of the diffraction pattern. So I'll play that video one more time. So you can see lights on, nothing's happening, and then suddenly diffraction pattern changes, basically expands. That corresponds to the lattice contracting in real space. So it appears as if 2 time scales are governing the transformation, because what we did is turn the light on, and then if we zoom into any of these diffraction points, you can see there's a period of time where nothing happens, right? The light's on and the system just kind of stays in its hydrogen-rich beta phase. But then we can start to see the splitting of the diffraction pattern where there wind up being 2 peaks, the beta peak and the alpha peak. So basically beta and alpha are coexisting until the system has basically fully transformed. So what we assign these time scales to is, first of all, just an induction time, where basically once the light is on, takes a little bit of time for the system to act upon that. And basically that light causes the release of hydrogen from basically the subsurface and very edge of the nanoparticle or tips of the nanoparticle. But then for all of the hydrogen to leave, you need to get the hydrogen from the center of the nanoparticle also out to the edge, right? So that basically is your diffusion time. So you need your hydrogen to hop from site to site within the nanoparticle. So when we vary the wavelength of light, what we found is that the induction time was very dependent upon the wavelength of light and how that wavelength of light corresponded to the plasmon resonances. So we collected the plasmon spectra of, say, one antenna reactor pair and found that actually had two peaks in this particular case, like a dipolar and a quadrupolar type mode. And those two peaks corresponded to where we had dips in the induction time. So the reaction was at least 10x faster when we were on that plasmon resonance. And I don't think I included it here, but basically the diffusion time remained constant, which is kind of what you would expect. Like basically for diffusion to occur within the nanoparticle, it doesn't really matter if light is there or not. And that also was kind of an indicator that there's not so much heating going on because we're not expanding the lattice and making diffusion faster because, you know, diffusion is thermally activated.
May I ask?
Yeah.
So this favorable wavelength is because of absorption or not? Because you just said it's not because of heating. I would expect that the material absorbs a lot there.
Yeah, great question. So in this case, yep, the plasmon spectra is a combination of absorption and scattering, right? So this is kind of your extinction resonance. So there is some local heating. When people have tried to determine the heating through a combination of computational and experimental approaches, they found that very likely the heating is not more than a few degrees. So that's not enough basically to cause a substantial change in the diffusion. And some of the experimental techniques that people have used would include SIRS, like people have done Raman scattering, looking at the ratios of the Stokes to anti-Stokes peaks in Raman, like Renee Frontera at, I think she's at Minnesota, has done some really cool work basically extracting the temperature. So even though they're strongly absorbing, you're not causing significant heating, in part because of basically the particular mode. So in this case, the mode is kind of centralized in the gap between the nanoparticle. Hopefully that helps.
If it's not because of absorption, then what causes these two wavelengths are the most favorable wavelengths?
Yeah, great question. So, you know, I'd say that's an area where hopefully I'll try to give you some experiments that point to what the answer is, but in this case, and in this particular paper, we had two hypotheses. So one of which is the light, is being absorbed, but instead of giving rise to heating and expansion of the lattice, it gives rise to those hot carriers that basically get quickly ejected or extracted from the nanoparticle and put into basically the LUMO of that hybrid hydrogen-palladium orbital that forms on the surface of the nanoparticle. So that's kind of one thing that's happening. So there's not so much lattice heating in phonons, but there are hot electrons, so hot carriers. And then another thing that's happening is because you have this increased electromagnetic absorption, you basically can have increased rates of transitions of light from the HOMO to the LUMO in that hybrid palladium-hydrogen orbital. So there's both direct electron transfer into the HOMO that can give rise to kind of hydrogen formation in the environment in this case, or you can also have basically increased rates of transitions that comes from the increased local density of optical states. Yeah, go for it.
So this near field enhancement, does it match with hydrogen's HOMO-LUMO? It does?
Yep. Or it matches with the HOMO-LUMO of, it's not just hydrogen, basically it's hydrogen bound or adsorbed to palladium. So people kind of call this like a hybrid orbital between your palladium, kind of D and S electrons, and your S electron of your hydrogen. So basically it matches with the HOMO-LUMO of that hybrid palladium-hydrogen orbital.
And that is normally much lower compared to free hydrogen, right?
That's right. Yeah. So what we found not only is that we have this expedited rate, but also that when we looked at different antenna reactor pairs, rather than having kind of this kind of random nucleation of hydrogen, that more often than not we got hydrogen nucleating at that electromagnetic hotspot, or basically hydrogen leaving the nanoparticle at that electromagnetic hotspot. And you can kind of see that in some of those contrast images that I show here. So if we look at a catalytic nanoparticle, we have various catalytic kind of hot spots. So basically where the catalysis wants to occur is generally those low coordination number sites. And here, for example, is gold 147, so 147 gold atoms on the surface of a catalytic nanoparticle. So the catalytic activity corresponds with the electrostatic potential, and that's highest at the tips. What we wanted to know is, could we use plasmons to make a site that normally is not the preferred catalytically active site active, and in fact the more preferred site for catalysis. So for example, could we imagine making the whole face of the nanoparticle catalytically active? So to test that, we looked at this crossbar geometry where we had a gold bar that was basically our absorber, and now a palladium nanorod. And here in cross-section you can see the electromagnetic hotspot is basically between the gold and the palladium. And I showed you earlier that hydrogen always wants to leave and enter from the tips of the palladium nanorod. But with this electromagnetic hotspot, we wondered if we could get palladium to leave or enter by the faces of the nanorod where we had this hotspot. So what we did is use electron beam lithography to pattern basically gold across the surface, and then we drop casted our palladium nanorods that were colloidally synthesized on top, and then we could map out the plasmon resonance, which is basically in the red, and you can see that visually in the electron microscopy image, right? Like the gold is looking this kind of vivid orangish reddish color. So here's the movie, and just to maybe talk you through experimentally what's happening, we're imaging one facet of the palladium nanorod. It's the bottom facet that basically is closest to that gold corner. And when we turn the light on, which happens now, you can see hydrogen left basically from the center of the rod and then went out both sides. And then when we turn the light off, you should, yep, now you'll see the hydrogen starts coming back in from the tips of the nanorod. And then it starts moving across the nanoparticle. So this bar that you see here in the middle is basically where we have gold. So we're not able to image what's happening directly on the gold because the electron beam just gets too scattered or too diffused by it. But we found that as we changed the illumination wavelength, we either got no nucleation when we were at lower powers, or we got middle nucleation when we were at the right power and at the right wavelength, basically right around those reddish orangish wavelengths to match with the plasmon resonance of that particular gold palladium dimer. And then we did some simulations basically to figure out which configuration was the least energetically favorable. And it turns out middle nucleation is the least preferred. Basically, it would take kind of the most energy going into the system to have hydrogen want to leave or enter from the middle. And that happens regardless of the length of the nanoparticle. So when your plasmonic or palladium nanorod gets longer, like having middle nucleation is a little less energetically unfavorable, but generally always the hydrogen would want to leave from the tips. But with the light, basically we have enough energy to overcome the barrier for these more atypical reaction pathways. And just to kind of put that in a little bit of perspective, if you think about the catalytic surface and your molecule nearby, you know, generally we're used to drawing out kind of a reaction coordinate that has kind of a potential well, and then we can excite this thermally to access different states within the potential well. But when we come in with the light, we can generate hot electrons in addition to phonons that basically give rise to an excited state, potential energy surface, that basically causes kind of a new reaction pathway that's not possible when we're just in thermal equilibrium. Okay, so what we want to do is link that to reactor scale performance. And the three reactions that we've looked at so far are acetylene hydrogenation, ammonia synthesis, and then kind of green steel making, or like iron reduction, where we use hydrogen basically instead of carbon dioxide or carbon monoxide. So here's a picture of the reactor we use. It basically is a light source that's coupled through an objective down to a reactor, and the reactor is basically small, probably about like this big. It's made of stainless steel, and it basically gets packed with the nanoparticles that we make. And if you look to the industrial catalyst that's used for acetylene hydrogenation, it usually is a silver-palladium alloy. And that's because palladium, as we saw in the previous slides, hydrogenates, right? So hydrogen wants to go in. But the industry has said, well, we don't want it fully hydrogenating because that means we're not going to have enough hydrogen to be able to do the hydrogenation. So they alloyed in some silver. And I think this is kind of neat because they weren't even thinking about the plasmonic properties. They just kind of alloyed it in to stop the hydrogenation. But if you put too much silver in there, then you sacrifice the rates because silver is inactive for hydrogen splitting. So here's the curve now that you've seen a couple times where basically the concentration of silver and palladium gives rise to this trade-off between conversion and selectivity in your reaction. So basically we hypothesized that by using light, we could give rise to the selective hydrogenation of acetylene, giving rise to ethylene basically when you need hydrogen. But when you don't need hydrogen, you can basically turn the light off and prevent ethane formation. So the idea is that with no light, there's hydrogen in the environment, and basically there's hydrogen that now is in the subsurface of your palladium or silver palladium bimetallic nanoparticle. And then with illumination, basically you can trigger the hydrogen to come out of the surface and subsurface to be used for your catalysis. So we focused on these silver palladium prisms. And again, we could do essentially atomic scale imaging in these reactive gaseous environments. And what we did is synthesize different bimetallic nanoparticles that have different concentrations of silver. And with our TEM, we can map out with close to atomic scale resolution where the silver is and where the palladium is. And then we can scan the electron beam basically over each position within our nanoparticle and map out the plasmon resonances, so we know exactly where light is going to be absorbed in the nanoparticle, not only spectrally, but also spatially. And what we found is that kind of, as you would expect for the prism, we have the bulk plasmon mode, we also have an edge mode, and then we have this kind of tip mode, where the light gets concentrated along the tips of the nanoparticle. And we can look at basically maps of these modes. So here we have just 25% silver. As we vary the amount of silver from 25 down to lower amounts, you get, as you would expect, kind of a nice shift in the plasmon resonance. And then we can monitor how hydrogenation is occurring by basically looking in the diffraction plane. We put our objective aperture over one of the peaks, and then basically that peak is going to leave our objective aperture when there's a lattice contraction that gives rise to an expansion in the diffraction plane. So here is our nanoparticle, and when we have no illumination, basically hydrogen will enter from any of the tips. When we're illuminating it, here we can follow basically how hydrogen is leaving the surface. As we'd expect, the transformation speed basically is pretty fast, like in this case over 30x increased when we have light. And when we compare basically the nucleation site with no illumination versus with illumination, Here we basically get nucleation occurring generally at this tip. So they're not all the same for this particular prism. This tip was a little bit kind of more like pointy. Whereas here we get basically nucleation occurring at the top. And when we do full field simulations that take into account the exact structure of the nanoparticle, we wind up finding that this peak is the one that has basically the strongest electric field enhancement, and that basically is what's driving the dehydrogenation. So we can follow that basically over multiple like hydrogenation steps. So in the dark, basically we wind up finding that the nucleation is pretty much evenly distributed to occur at kind of that middle radius of curvature. Whereas in the dark, so yeah, basically, in the dark, the number of nucleation is almost evenly distributed across like the smallest radius of curvature peak, the middle radius of curvature like tip, and the maximum radius of curvature tip, so almost an even distribution. Whereas when it's illuminated, basically you're always at that sharpest tip where you have kind of the strongest electromagnetic field enhancement that you're getting most of the nucleation. And then what's cool is we can also see how we get different reaction mechanisms. So here I think you'll be able to see hydrogen leaving the nanoparticle in the dark. So you can see this cool like interface now with much higher resolution than we've had before. And basically the hydrogen then leaves out of both of those tips. So you saw it kind of moving across the nanoparticle and then splitting. Whereas with illumination, basically it doesn't have that same split. So I'll play that video again. So here, much slower, hydrogen's moving across the nanoparticle, and then eventually it winds up kind of splitting as it gets these two tips, forming two distinct phase fronts, there and there, that then leave out of the tips of the nanoparticle. Whereas with illumination, it's not only much faster, but it basically has this like almost rapid rotation that makes it just go out one edge of the nanoparticle. And then my colleague Felipe Hornada has shown that when you have these single dopants, like single metal atom alloys, you don't wind up changing the plasmon resonance. So in fact, whether you have just silver or silver with one palladium atom, your plasmon resonance is the same. You can really only see a difference if you start doing the subtraction between, say, a silver 55 cluster and a silver 54 with one palladium. But you do wind up getting this cool change in the charge density. So when he does these excited state calculations, you can start to see, you know, some of these theories that we had about hot carriers coming to fruition, where you don't change the plasmon resonance, but your charge distribution gets more centered over where you have these catalytic atoms when you are illuminating the silver nanoparticle. So you basically get hot carriers that get distributed over your catalytically active material. Cool. So what we do is we make nanoparticles, and here you can see as you're varying the weight percent of palladium in silver, you can get a pretty vivid color change from kind of this pinkish reddish color all the way to like a very dark burgundy. And then what we do is we put them onto an alumina support. So this is basically almost like a sand, like grains of sand that you put your nanoparticles onto. So here you have this nice powder that starts to look a little more like purplish. So here are some of the nanoparticles. They're the colloidal bimetallic nanoparticles. We put them onto the support substrate, which is kind of like grains of sand, and then you pack them into that reactor that I showed you earlier. And we know now from the TEM basically like, you know, where the bimetallic nanoparticles are located, where they have their plasmon resonances, and also how much hydrogen gets loaded into them when we've tuned the composition of the nanoparticles. So we have a reactor, light comes in, and then we also have a gas flow system that introduces gases into the reactor, and then we can control the temperature of the reaction in addition to the illumination conditions like the wavelength and the power. And then we have a gas chromatograph basically to read out what products are coming out. So basically up until now we've only really looked at the hydrogen side of things, but here we finally get to look at how that subsurface of hydrogen is going to impact the products that we get. And then my student Briley has written this really neat automated software for controlling the photoreactor and also for analyzing the data that's coming out. So if any of you do catalysis and have a setup that basically looks like this, feel free to go to our GitHub site, so github.com, dionelab/catalyte, and hopefully we'll make a lot of your reactions much easier. Like now he's streamlined things where he can go and enjoy, you know, spring break or a long holiday and basically be running thousands of reactions. So I showed you a plot earlier from Dane Swear where he was able to look at acetylene hydrogenation both with heat and with light. And he was able to get kind of this interesting difference between selectivity and conversion with both of those. But with some of our more compositionally optimized catalytic systems, we're able with light basically to get not only high conversion of the acetylene to ethylene, but also very high selectivity. So we don't have very much ethane that's being formed in this reaction. So you can see the conversion and the selectivity when you illuminate on resonance in these silver palladium bimetallic alloys is well over 80%. And then what we did is change the mass of the catalyst. So in photocatalysis, what we found is that when you change the mass of the catalyst, basically as you increase the illumination power of the blue laser that we were using here for photocatalysis, basically the conversion yield just keeps going up with increasing power, and it doesn't really matter how much catalyst we're using. Whereas in thermocatalysis, basically as you reduce the amount of mass of the catalyst, you basically wind up getting less conversion, and that kind of makes sense. So basically the less catalyst you have, basically the smaller your conversion rate will be in thermocatalysis because you're pretty much heating the entire reactor bin. Whereas regardless of whether we have 3 milligrams of catalyst or 20 milligrams of catalyst, we don't really change the conversion, and that's because the light is pretty much only heating those top few millimeters anyway. So this kind of points to when you're thinking about photocatalysis, how you can design a reactor basically to have LEDs kind of all around your catalytic material so that way you're uniformly heating things. And there's a company that came out of Rice University called Syzygy that now has designed really cool photocatalytic reactors that allow for very uniform illumination. And then by tuning the amount of silver and palladium, we can basically tune whether we have a plasmonic effect or a photothermal effect. So here on the vertical axis, we're looking at the conversion of acetylene to ethylene. And as we change the temperature, you can see that basically with less catalyst, this is a plot you saw on the previous slide when we have a large amount of silver and a very small amount of palladium in each bimetallic catalyst, that the conversion goes down. But when we have basically a large amount of palladium and a small amount of silver, even though the conversion is a little bit lower with less catalyst, it basically remains almost constant with temperature. So here we're able to keep the conversion yield almost constant. But the cool thing is, like with photocatalysis, again, you saw this plot earlier, here we have basically illumination that is giving rise to higher conversion yields and higher selectivity. But when we basically get rid of that plasmonic response by having a lower amount of silver, so almost no silver in our system, now we get a response that looks almost more like the thermocatalysis response, right? So down here, the laser is basically inducing just some heating, not any plasmonic effects, whereas up here, the laser is also inducing hot carriers that are concentrated around those palladium atoms that are giving rise to increased conversion as we turn up the laser power. So I'm nearing the end, but I just want to give you a couple more examples. This one is on ammonia synthesis. So here we have hydrogen plus ammonia, or plus nitrogen, that's giving rise to ammonia. And basically the standard catalyst that's used is an iron ruthenium system. But my postdoc Lynn was able to basically swap out the iron for gold, so that way he could work with these more plasmonic responses. And when he controlled the amount of gold, he could get nice like plasmon resonances here in the green that experimentally agreed quite well with what he simulated. And then he also controlled the phase, so he had FCC nanoparticles and HCP nanoparticles. And then when he put those into the reactor, what he was able to find is that with illumination, he was basically, for both the FCC AU, RU 30% ruthenium catalyst alloy and the 20% ruthenium alloy, basically get a response that kind of mimicked basically the hot electron cross-section that he had calculated. So you can see there starts to be a peak in the ammonia production in this particular case. And what I think is really cool is that he was just using 100 milliwatts for his laser intensity. And he was at room temperature and pressure, whereas normally for these iron ruthenium catalysts, you need about 20 megapascals of hydrogen pressure or gas pressure and 500 degrees C. So pretty cool that he was able to make ammonia basically at room temperature and pressure without any sacrificial reagents. You can see that the quantum yields are pretty low, like less than less than 1%. But I think it's pretty encouraging to think about how we can optimize this process to be able to have more sustainable ammonia production for fertilizers by using some of these illumination properties. And kind of a cool thing, many of us have nitrogen in our bodies. Most of it is in our proteins. And over half the nitrogen in our bodies came from a Haber-Bosch process, which basically is the industry standard process for making ammonia. And that's because basically ammonia is needed for fertilizer. The fertilizer goes into agricultural products, and then obviously we eat the food. So over half the ammonia in our bodies basically was enabled by this particular high temperature, high pressure catalytic process. But I think it's neat to think about how we can reimagine the Haber-Bosch process to be more sustainable by using illumination. And then just the final example is how we can use some of these bimetallic systems for steel manufacturing. And steel and cement both account for about 8% of all global greenhouse gases in kind of materials manufacturing. And I don't know if any of you have seen some of these like furnaces where they make steel. Basically, they'll input iron ore into this huge furnace, and like for scale, like the size of the human is about like the size of my mouse down here. So these are like really enormous systems for steel manufacturing. And basically, they'll heat them up basically to temperatures of a few thousand degrees C to try to go from Fe2O3 to pure iron to be able to create steel. So usually they'll use CO to be able to reduce the iron ore. So Fe2O3 plus 3CO gives rise to the iron that you need to make steel, but then you also emit a lot of carbon dioxide in the process. And this is an exothermic reaction, so it's thermally favorable. But like I said, emits about 8% of all global greenhouse gas emissions. So my colleague Leora Dresselhaus and also Xiaoling Zhang and Felipe and I have been trying to think about how we can do direct iron reduction with hydrogen. So now you have Fe2O3 plus hydrogen. That gives rise to the same iron that you need, so 4 Fe. But instead of giving rise to greenhouse gas emissions, you just get water in the process. The challenge is that it's an uphill reaction. So the idea was like maybe we could use light to be able to drive this reaction. So here we started looking at reduction of Fe3O4 with hydrogen and also with illumination. And you can see that in this TEM video, we went from basically not seeing anything to now seeing these like darker images that basically correspond to some of the pure metal that's being formed, basically as this like uphill reaction is occurring with illumination. And I have some images here where you can see there's actually a lot of parallels to some of the hydrogenation we saw in palladium, where it looks as if hydrogen is basically entering from the tips and then forming these sorts of phase interfaces it and moving across the nanoparticle as the Fe2O3 basically becomes pure iron. Okay, so that's most of the science. Here's kind of what I think the future holds for plasmon catalysis. You know, first, I think there's a lot of exciting opportunities in developing a predictive theory of plasmon catalysis, and it's kind of a complicated area to think about, in part because we have really good theories that work on the bulk scale, where you have like a perfectly periodic lattice, and we have really good theories that work when you only have a handful of atoms or kind of like molecules or macromolecules. But catalytic nanoparticles are in this interesting middle ground where you have hundreds to thousands to tens of thousands of atoms. So it's not quite a fully periodic structure, but it's so large where, like, up until recently, it's been really hard to do a lot of first principles calculations of what the excited states are. But I think with advances in both like excited state models as well as in kind of supercomputing capabilities, we have the ability to do these excited state calculations. So figure out what happens to the phonons, photons, electrons, excitons, etc., and plasmons in these multimetallic nanoparticles when they're illuminated, and basically map out plasmon dependence on size, shape, and dopant concentration. And then also be able to excite the nanoparticles and map out those excited state potential energy surfaces, which, you know, up until now I just showed you schematically, but I think there are advances in computation that are allowing us basically to map out what those potential energy surfaces look like in a more predictive way. And then I think, as you saw in this presentation, we're only kind of at the tip of the iceberg for being able to link the atomic and molecular scale properties of these photocatalysts to the reactor scale performance. And what we're excited to do is link kind of how the various like dopants at specific positions influence both their electronic band structure and their plasmonic responses, and then also develop spectroscopies to kind of probe these molecular transformations. And we only really probe the hydrogen in the TEM, but we're starting to move to the point where we can introduce additional gases and kind of follow with vibrational spectroscopy what is happening to the molecules in the environment surrounding the catalyst in the TEM, and then also put that same vibrational spectroscopy capability into our bench scale reactor, so we can follow with both high spatial and high temporal resolution how the catalysts are basically giving rise to molecular transformations. So with that, I'll summarize by saying I think you know, plasmons are kind of an emerging area to be able to control catalysis. In most of the work we do, we excite plasmons with lasers, but there's no reason why you couldn't use either sunlight-driven LEDs or direct sunlight to be able to excite catalysis. We were able to use a technique that we developed over the course of a decade, optical excitation within a TEM, to be able to understand how light can basically selectively trigger the release of hydrogen, giving rise to product selectivity. So you basically have hydrogen on demand. So you can use the nanoparticle as a sponge to absorb hydrogen, but use the light to trigger hydrogen being released from the nanoparticle for selective hydrogenation. And then when we were in the dark, we saw that single crystals basically, like, you know, fully absorb hydrogen, so they don't exhibit thermodynamic phase coexistence. So they allow for maximal energy storage, so to speak. So there are a lot of parallels with lithium-ion batteries there as well. And then I also showed you how light enables catalytic site selectivity and increased reaction rates, and also allows for selectivity in the conversion by being able to trigger the release of hydrogen. So with that, I'll just put up a picture of my lab. This was at a barbecue not too long ago. And I think we're about 30 minutes away from the end, but happy to take any questions, whether on this topic or anything in general, if you just want advice on, like, you know, being a scientist or career paths. So thanks so much for your attention and for dealing with my jet lag too.
I would like to kick off. First of all, thank you a lot for this very informative, clear, and moreover educational lecture. Especially it is very helpful for an outsider of chemistry like myself. That this top-bottom way of pitching is really helpful for a person like me. So knowing the fact that I'm an outsider, so I will not be able to deliver very specific questions. Instead, I would go a little bit more into general. The question from my side is, how would you evaluate the energy efficiency of such a new this new approach, and knowing the fact that what we do here is actually using the plasmonics to manipulate more, in particular to reduce the energy barrier or to guide certain reactions into our favor in the case, for the sake of reducing the energy consumption in the conventional approach. But also knowing the fact that this plasmonics were, in the end of the day, used as a local heater, and generating heat from a coherent light source is by definition not very energetic efficient. How would you evaluate such access the energy efficiency of such approach? What is the state of the art?
That's a great question. So I'd say the main way you can evaluate the energy efficiency of either is either by thinking about thermal energy, right, as being kBT versus photon energy, h nu. So you need to do kind of a one-to-one correspondence to figure out what is the kind of energy cost per photon. And as long as you can use kind of solar-driven LEDs or direct sunlight, the energy is actually, or energy efficiency can be like orders of magnitude higher than thermal catalysis, and you're not relying on heating, right? So that's the important thing. So the illumination is not necessarily driving local heating of the nanoparticle. The illumination is giving rise to excited electrons in your metal nanoparticle, and those excited electrons are giving, or basically driving, the chemistry. So I think the field has converged around like many experiments showing that it's basically the hot electrons and hot holes that are driving the chemistry. And now kind of the task ahead is to figure out how to design nanoparticles that get you the highest concentration of hot electrons and hot holes to get the most energy efficient reactions.
Yeah, amazing presentation. Thank you.
Oh, thanks.
I actually had a couple of more general questions. So, for instance, you were talking about already some reactors that have been developed where they uniformly illuminate the catalyst. But if we have to scale this up, how would you ensure that it also reaches the middle, for instance, of the center of the reactor?
Mm-hmm. Yeah, really great question. I think the reactors that are being developed for kind of plasmon photocatalysis are being inspired kind of by flow reactors that often are used, I think, in like the pharmaceutical industry, where you can think about kind of a thinner tube, like a thinner capillary tube that gets packed with your catalytic material, but then kind of wound almost like a coil or a helix, and then you can wrap basically around that coil like LEDs all around it. So you still wind up uniformly illuminating your entire catalytic powder that basically is in your reactor, but then you wind up getting as much material as you would have kind of packed just into a standard reactor.
I see. Okay. Sounds challenging.
Well, a little bit. I don't know if you've ever seen like a liquid chromatography setup. It basically is like almost like a fiber that then gets packed with little beads, right? So it's similar to that. It's basically a fiber that now gets packed with your catalytic bed. And then they just have like a kind of, I think they're almost like 3D printed LEDs that just get kind of taped around the helix. So I'd say, from what I've seen of the company that's making these, Syzygy, like at least in the United States, they've done really well. Like they're not a public company yet. They're a private company that's funded by like venture capital funds, but I think they're at their like Series B raise at this point, and they have quite a few like big customers internationally. So I think, to your point, seems challenging, but also one where for the particular reactions they're looking at, which I think is creation of like green hydrogen, it actually winds up being like not only more sustainable, but also there are good economic incentives around it.
I see, I see. Yeah, I can ask another question, I guess.
Sure.
Are there any plans for light outcoupling in a TEM? Do you think about that? Dream about that?
That's a really great question. So the holder that we developed with Gatan has 2 parabolic mirrors and then 2 fibers. So we can do light in or light out, or light in and light out from each of those fibers. So we've done some cathodoluminescent studies on nanodiamond. I had a student who was really excited to look at silicon vacancy centers in diamond. And I had another student who was looking at 2-dimensional materials like hexagonal boron nitride and color centers in those. And there we basically used the electron beam as kind of like a white light source, and then could couple light out to be able to understand how the optical properties correlated to the atomic scale structure.
So you have it already, basically.
Yep.
Nice. Yeah, last question, and then I give the mic away. Any hurdles to watch out for when doing light coupling into a TM? What is the, well, do you have any tips, let's say, when you want to do that?
Yeah. You know, I guess first thing is just doing accurate mapping of how much power is going to which part of the sample. Like I'd kind of call that almost hotspot mapping. Like if you're doing light incoupling, just knowing like where the light is hitting and making sure that's aligned with like where your sample is, because you know these holders are a little bit finicky. So that's one thing to watch out for. Also just know that the amount of light that gets coupled into the fiber and then subsequently onto your sample is just a small fraction of what you put in. So, you know, don't buy just a 1 milliwatt laser and hope that you'll be getting milliwatts at your sample. You'll usually be getting much less. And then I'd say if we had the chance to redesign a holder all over again, having like polarization maintaining fibers would be really neat. And I know some of you who maybe do SEMCL like already have that capability, but being able to do polarization mapping is really cool. Also like momentum mapping, that's really hard to do in the TEM, and something where if I were to, you know, have 50 more years, or maybe not quite that long, but I think it'd be really cool to develop a holder that allows for that momentum mapping or like dispersion mapping in the TEM. But I think it probably would require more than a holder design. You'd probably need to design your own TEM to be able to do that. So that's why I gave it such a long time horizon. I'm probably not going to build a TEM, but some of you might.
Okay, thank you.
Thank you for the super interesting talk and very clear talk as well. But I do have a question regarding this presentation. So I understand, like through plasma photocatalysis, you go to more localized and faster reaction, and in the end you get higher selectivity in the reaction. But how exactly the local plasma effect actually increase the selectivity? Presumably by avoiding over hydrogenation of the reaction. But I'm lost a little bit in the middle of the part.
Cool. No, it's a really good question, and this is our best guess as to what's happening. So I'll try to show 2 pictures. So first, let me go back to what the reaction is that we're looking at. Okay. So we'll just focus on the acetylene hydrogenation for now, because that one I think is a little easier. So you have the C triple bond C with 2 hydrogens. Then you have hydrogen in the environment, and you're trying to split that hydrogen and add it onto the ethylene. But you don't want to fully hydrogenate it and get the fully saturated ethane. So if now I go to this picture, what we have found is that when there's no light, basically hydrogen wants to be absorbed in the palladium. But when we illuminate on resonance, we can basically release that subsurface hydrogen. So you can think about using the right wavelength of light to basically get the hydrogen out of the surface to hydrogenate your acetylene, but not over-hydrogenate it, right? So you can basically then turn the light off to get the hydrogen to go back into your system.
So you need external intervention during the process. So
You know, right now we haven't, but basically with illumination, we're able to, you need illumination at the right wavelength, right? So we basically illuminate at the right wavelength to get enough of the hydrogen out of the surface. If you're pulsing it, I think that's actually a really cool idea that we haven't tried yet, but you can think about kind of triggering the light on versus off to be able to kind of more precisely control both when the hydrogen is being illuminated and how that's corresponding to different either electronic transitions or vibrational transitions.
So now the light you use is like a continuous light, but it's also laser, right? It's
Yep, it's just a laser.
okay.
I had two questions, but really nice talk. I actually don't do this kind of catalysis, but I do more electrocatalysis. So one of the problems that we have commonly in electrocatalysis is that the catalyst keeps changing, and then the conversion efficiency in general just goes down. So I wanted to know if you also have the similar problem in gas-solid interfaces.
Oh my gosh, such a great question. I didn't show the results on kind of CO2 reduction. I don't know what sort of catalysis you're doing, but there we've had a lot of issues with the catalyst changing. With these catalysts that we've done the acetylene hydrogenation on and the ammonia synthesis on, the catalyst is quite stable, and what we've done is kind of try to optimize which support substrate we're looking at. So in this case, with the acetylene hydrogenation, the silver palladium on the alumina support seems to minimize like sintering. And the catalyst nanoparticles, at least before and after catalysis, seem pretty much the same, but I think there's actually a big opportunity to look at these in the TEM, basically, and follow like how maybe single atom alloys are like migrating around the surface. So we haven't done that yet, but at least it doesn't seem like there's a change in activity with time for these particular catalysts.
My second question was that you said dehydrogenation is much faster, at least from the videos that you showed, than the hydrogenation. So I wanted to ask you, if you do the hydrogenation the second time on the same catalyst after dehydrogenation, is it faster? Is it the same?
Oh, yeah, that's a really great question. So basically, I wouldn't say the hydrogenation versus dehydrogenation is faster or slower. It's always, whichever of those processes you're looking at, it's roughly about the same period of time for each, but it's always faster with the light on. So I think the examples I showed you are light on with hydrogenation versus dehydrogenation. So the light is what is expediting the reaction. And what we do in all cases is cycle the system basically 3 times with hydrogen to make sure that like any lattice imperfections during the colloidal synthesis have kind of relaxed. So we usually find that the results, once you've cycled it 3 times in the TEM, are very consistent.
I have a couple of questions. Thanks for the nice presentation. My first question is that regarding the conversion of the ethine to ethene. So in that one, is it a fixed bed reactor or is it a continuous flow reactor?
Yeah, this one is essentially a continuous flow where we're just using basically a Herrick cell reactor, and then flowing the acetylene and the hydrogen continuously over the reactor bed, and then monitoring the products with the GC.
So how do you optimize the light illumination and illumination part so that the selectivity increases? How do you, like, what factors do you take into account to optimize this selectivity?
Yeah, so for the illumination, we're mostly varying, so you can see it basically gets coupled in through an objective here. So what we can control is, first of all, the power, right? Just changing the power of the laser itself. And then we can also tune the wavelength. So we have a super continuum white light source where we can illuminate basically on versus off resonance of the nanoparticles when they're on the alumina support substrate.
So do you check that? Do you check like a certain percentage of ethine has been already converted, and then you illuminate?
That's right. Yep.
Okay,
That's
okay.
right. Yep.
And another thing was, why does the hydrogen leave from the tip of the nanoparticle?
Oh, yeah, that's a great question. So basically, if I go back to this slide, for most catalysts, like the catalytic activity is always the highest at these low coordination number sites. And those just wind up being, at least for the catalysts we look at, the most kind of preferred atomic rearrangements to have hydrogen split and then kind of absorb onto the palladium itself.
Okay, and as it's the most reactive, it also dissolves the hydrogen faster.
Exactly.
Okay.
Yep.
And the last question is, what is the role of the hot holes present in the system?
Oh, that's a really great question. So we've thought mostly about hot electrons, but in certain systems like a CO2 reduction, you'd probably wind up using probably a gold palladium system. And gold has interband transitions, and those can give rise to hot holes that are almost more important in driving catalysis than some of the hot carriers.
Okay, thank you again. Another outsider's question. Actually, my first one was almost identical as the previous person who asked. Previous person. My question was actually regarding the many holy grails you mentioned at the very last of your presentation or lecture. That is to use non-coherent light or the LED or the natural light to excite what type of plasmonic-driven reactions. How is that possible? Or are you actually talking about the plasmonics at all?
No, it's a great question, and our lab hasn't done it, but there have been labs that have just used like LEDs or solar-driven LEDs. And just to put things in perspective, if you've ever seen a stained glass window or a rose window, like that does not need to be illuminated with a laser to see the colors, right? And basically those stained glass windows are just metal nanoparticles doped into glass, and the resonance of those metal nanoparticles is what's giving rise to the color you see. So very easy to excite resonances in metal nanoparticles. And we just use a laser because it's a more convenient source to make sure we have that wavelength tunability.
That actually impose certain, how to say that, the particle need to be by itself geometric.
Exactly, yeah. So the nanoparticle itself has resonances that are due to its size, shape, composition. And those give rise to the kind of vivid optical scattering you'll see. So if you look at a bright field or a dark field image, the nanoparticles will scatter different colors.
I see. How to be spherical geometric. Symmetric. Would that limit how much you could do with plasmonics?
Oh, so they don't need to be spherically symmetric. And in fact, you can have, say, nanorod geometries. And there you have two different resonances. So the color you'll see will be dependent upon either the polarization of light or if they're all aligned. Or you could just illuminate with like a filter, right? If you have sunlight and then you put a red filter in or a green filter in, that might correspond to, say, the longitudinal resonance or the transverse resonance.
Yeah, hey, thank you so much. It was super nice. I had a more fundamental question about what's exactly meant by hot electrons and hot holes?
Oh, yeah, really great question. Thanks for asking that. And I probably skimmed over it too quickly. This picture is not the clearest, but okay, so let's see if I can find my mouse. Okay, if we're down here, if we think about this as the vertical axis as being energy and the horizontal axis as being like probability, if we look at the Fermi-Dirac distribution function, basically the occupancy probability at t equals 0 is kind of the step function, right? So basically the Fermi level is where you have an occupation probability exactly equal to 1/2. And then you probably know the Fermi-Dirac distribution function starts to look a little bit more sloped as you turn up the temperature, right? So basically you get more thermally excited carriers. What can happen with illumination is even if you're at 0 Kelvin, illumination, without changing the heating, can give rise to electrons that are in higher lying, previously unoccupied states by virtue of that photoexcitation. So you get a Fermi-Dirac-like distribution with electrons that are above the Fermi energy, but it doesn't come from thermal excitation. So that's what people refer to as hot electrons. They are basically electrons that are hotter than the distribution that you would expect just based on the temperature of the metal.
And what's then the hot holes part of that?
Yeah, so basically the hot holes comes from normally, like when you think about a metal, you just have a parabolic dispersion diagram, right? Like E is proportional to K squared. And then you can draw your Fermi energy as where you have like all the states, below are filled, all the states above are empty. So light comes in and then it starts like exciting electrons from below the Fermi energy to above the Fermi energy. In gold, you have this kind of parabolic dispersion that corresponds to your S band, but you also have an interband transition that's like an S to D band. So light can come in and excite carriers basically from the D band to the S band, and then your D band has hot holes that also now can contribute. So basically those are, holes that are in your D band. So if you think about the maybe analogy to a semiconductor, right, where you have a valence band and a conduction band, you can have conduction electrons. They contribute to your conductivity, but you also have holes in your valence band that also contribute to your conductivity, right? So your overall conductivity is the sum of the concentration of your electrons, times their, conductivity, and then you also have to think about the hole contribution. So it's the same thing for kind of those interband transitions in gold. You have to think about the empty states that are left behind in the D band and how those can contribute to catalysis. Hopefully that helps.
Hello, thank you for the very great talk. I had a question about different modes where these light is being localized. So initially you explained plasma resonance, right, where the particle acts as a dipole, but when you showed these different modes. So in those cases, what's actually happening? It's not the dipole anymore, right?
Yeah, great question. I was trying to see if there's a whiteboard, but there is not one here. So, oh, it is. Oh, nice. Okay. Cool. Oh, yeah. So in, let's say, there are different sorts of modes that can occur. In, let's say, just a spherical nanoparticle, you can have a charge distribution that's like plus and minus, right? So that's your dipolar-like mode. You can also, I was talking about the nanorod before, you can have plus, minus, or you can also have plus, minus, right? If your electric field is polarized along the vertical direction. And then you can also have higher order modes, like a quadrupolar mode, so like a plus, minus, that's a quadrupolar type mode. And then same if you have like these nano antennas, you can have like a plus, minus, right? So this is kind of like a bonding dipolar mode. If you had a trimer, you could have like a plus, minus, kind of plus, minus. So this starts looking almost more like a rotating displacement current. So this is more like a magnetic dipole. So in metallic nanoparticles, yeah, dipole. Here's a quadrupole. You can have octopoles, yeah, heptapoles. So for the most part, we always have like electric dipoles, but there are certain configurations where you can also support magnetic like dipoles and higher order modes. And I guess the magnetic dipole is more similar to, you know, kind of this quadrupolar mode. So if you look at, it's called like Mie theory, named after Gustav Mie. So in the expansion of the electromagnetic modes of, say, a spherical nanoparticle, the quadrupolar mode is the same order as the magnetic dipolar mode. Does that help? Okay.
I had one more question. So
Sure.
you mentioned for CO2 reduction you were using gold palladium nanoparticle, right? But in the field of electrocatalysis, copper nanoparticles are commonly used. So then why did you choose to go with gold palladium?
Yeah, great question. So I say Naomi Hollis had some really interesting results of how using plasmon excitation of gold, she could get splitting of hydrogen. So it's kind of a neat system to be able to use kind of the combination of gold and palladium both for light absorption, but again also for some of that kind of controlled hydrogenation in the CO2 reduction process.
And my last question is, so in a slide you said that the quantum efficiency is less than 1%, right? For plasmon photocatalysis. Then, yeah, what's the future of plasmon photocatalysis then?
Oh, yeah, that was just for the ammonia production. For the acetylene hydrogenation, the conversion and efficiency are much higher, right? Like closer to or above 80%. For the ammonia production, it's so low, in part because those were results at room temperature and pressure with just 100 milliwatt lasers. So if you compare room temperature and pressure and only getting below 1% for ammonia, it's a really promising start when normally you need to be at 200 megapascals and like 500 degrees C. So we certainly could have turned the temperature up a bit or the pressure up a bit and probably gotten higher yields, but I think there's a lot more room to play around with the particular nanoparticle, like phase, geometry, composition, etc.
Thank you.
Hi, thank you for your talk. I have a question. Have you tried doing something similar, but not with metallic, but dielectric nanoparticles? Because like they are so-called cavity resonators, and they don't have, for example, ohmic losses. And in principle, it can be even better if you want to create some energy-effective process that,
Yeah, really amazing question. So on Wednesday, my presentation will cover more of the work that we've done on dielectrics, which are great for the reasons you mentioned. First of all, they support these Mie-like modes or Gustav Mie modes. They have strong optical absorption, in some cases similar to what you can get for these metallic systems, but there's no heating, which is nice for certain molecular transformations. The main reason we've mostly worked with the metallic nanoparticles is because they have generally more catalytic activity, so they're more chemically reactive than the dielectrics, which tend to not have the same sorts of tunability over kind of the site selectivity and the formation of those hybrid orbitals that you need for catalysis. So generally in metals, whenever you have D bands, there's a lot of cool chemistry that can be done. So I'd say, like, for the most part, chemistry is always done with D orbitals, which just makes it a little bit easier to use metals rather than with a semiconductor, you have all of your electrons just kind of locked up in your covalent bond, so it's harder to drive chemistry. And even with enzymes, usually they're metal-centered proteins that you'll find being like the most commonly highly active catalysts.
Thank you.
Yeah, but I will add on there, like, I think there are a lot of opportunities to combine the semiconductors with metals, and like it's a whole new ball game in terms of like not just using the semiconductor as the light absorber, maybe to drive chemistry in the metal, but you can also think about, like you mentioned, different resonator structures. You can think about like entering a strong coupling regime between the semiconducting cavity and the metal nanoparticle itself, and maybe even driving new new chemistry with like polaritonic states if you use dielectric cavities. So yeah, I'd say I wouldn't be surprised if that's an area that like pops up in the future as being a really exciting one.
True. Thank you.
Thanks.
I have a question. Actually, I don't know if it's a valid one, but I'm wondering, like, in the case when you have a platinum nanorod sitting on top of the gold, so when the reaction, the dehydrogenation reaction is going on, does that sitting on the surface of the gold change the plasma resonance of the gold over the reaction time?
Yeah, great question. So first of all, the palladium gold itself has its own plasmon resonance, like just by virtue of the coupling between the antenna and the reactor. We illuminate with, or basically the hydrogen like leaving and entering the palladium changes the bulk plasmon resonance, but it doesn't change the gap plasmon resonance. So the bulk plasmon resonance is kind of that metal to dielectric transition resonance, and that changes between about 6 and 8 EVs. But the surface plasmon resonance, or that gap mode resonance, doesn't change very substantially. In both cases, you can almost think about the either palladium or palladium hydride as being effectively equivalent from a dielectric or refractive index point of view when we're at visible wavelengths. So that gap mode remains pretty much the same. It shifts by a few nanometers.
Yeah, thanks.
Yeah, really great question. Yeah, in both cases, they're both metals at those frequencies. So that's probably the easiest way to think about heuristically why the resonance doesn't shift that much. Cool. I think we're at the top of the hour and they have dinner, so I'm happy to chat more over dinner. And like I said, happy to take more questions on the science, or if you just want to know what it's like being a professor or a group leader, or what California is like if you're thinking about postdocs, happy to share with you. Cool, thanks all.